Hey BioTeam. So you guys have learned that in order to build a protein, our cell will start with a gene of DNA. It will transcribe that gene into RNA, and then the RNA actually builds the protein in a process called translation. This video actually focuses on an intermediary step. Before the RNA goes on to build proteins, RNA gets processed. So after RNA has been transcribed, but before RNA is involved in translation, RNA goes through several modifications. And so before the RNA is processed, it's called pre-RNA. And after the RNA is processed and modified, it's called mature RNA. So there's two steps to RNA processing that we'll cover in this video. And the first of those you did in class today, and that's splicing. And splicing of RNA just means removing unwanted pieces from it and then stitching the remaining pieces back together. So if we have a strand of, let's say, messenger RNA here, let's say we have two chunks on either end of it that we want to use in translation. We call those pieces exons. And this piece in the middle that we don't want to use in translation, we call that an intron. So in order to remove an intron and splice these two exons together, our cell will use a series of proteins called spliceosomes. And what they'll do is they'll push the spliceosomes into the intron, which pinches out the intron and forces the two exons to splice together. So why is splicing important? Well, our cells can do something called alternative splicing. That is, they can splice the same strand of mRNA in different places, and as a result, they can produce different proteins from the same gene. So for instance, if this is our mRNA strand, and let's say that these codons in the mRNA strand code for polar amino acids, and these codons in the mRNA code for non-polar amino acids, well, it's possible, depending on how our cells splice this mRNA, that we could end up with an RNA strand coding for an all-polar protein. Or it's possible that we end with an mRNA strand that would code and translate to an all-non-polar protein, or it's possible that we end up with some sort of combination of the two. So after RNA has been spliced, a process called capping and tailing of the RNA can occur. And capping and tailing are exactly what they sound like. The process of capping is adding a 5' cap to the head of the RNA strand. And this cap is usually a guanosine triphosphate molecule. So very similar to an ATP molecule, which is adenosine triphosphate. Meanwhile, tailing of an RNA strand involves adding a poly-A tail. And that is adding a long series of adenines to the 3' end of the RNA strand. And what this does is that prevents enzymes from getting a hold of and digesting this RNA strand while it's out venturing in the cytoplasm, bumping around looking for the ribosome. And so that's it. At this point, you guys have some practice problems. We'll see you next class.